Urinalysis. Urine is usually the end product of kidney function, and in this experiment we're going to look at how to diagnose human being by using the urine sample. So the first part of urinalysis is to look at the color of the urine. So we have samples A to D, and you're expected to look at the colors and record your findings. So the color is very significant in terms of diagnosing if somebody is healthy or not. Then the second part is to look at the transparency of the urine samples. Look at the transparency and see what color you want to give. Is it cloudy? Is it transparent? And then you record that in your results. Then the third part of this is to look at the odor of the urine. So you have each sample, you smell them to say what kind of smell you can get from it, and then after you're done with that, you record your findings also. So we can also determine the specific gravity of the urine, and there's usually a normal range for that, 1.003 to 1.030. It's in your manual. So what you use is what we call the refractometer, and it has like a space in the front. So you take a drop of your urine sample, and 1 or 2 drops, and once you put that in, you cover this space, and then you can look through an aperture here, and it's going to give you the specific gravity. So the most important part of urinalysis diagnosis is to be able to determine different components of urine, and we're going to be using what we call the MULTISTIX test. This manufacturer is chemistry, and it has different parameters that you can look in. You can get the glucose level of your urine, the ketone level, you can get the blood, if there's blood in the urine, the protein or pH, and so many other things that you can find in urine that are usually abnormal. To perform what you use is the chemistry, which is like what you have to dip into the urine sample. So for example, you have for A, you can dip it in and then slant it a little bit to leave it at an horizontal angle, and then you leave it for 30 seconds, and after 30 seconds you take it out, and you can now compare with the readings that you have in here. So there's a reading here, and then you can compare what you have because there's a calibration of it here. Alright, so for the microscopic part of the urinalysis, what you need to do is to have a concave glass slide. You can also call it depressed glass slide. As you can see in the video, it has like a concave depression in it, and that's where you put your sample. Okay, so so now you just have to take out the slide and start. You put a drop of your sample in the depression area, and once you have that in the depression area, you had your cover slip on it, and then you can visualize this under the microscope at different magnifications. And if you're looking at something that like the red blood cells or white blood cells, remember you need to have the oil immersion and look at it at 100x magnification. So I'm going to look at this and look at it for all the samples that I have, and I'm going to put the results for you in the document I'm going to send to you.